Okay. Sige, so this afternoon we'll already begin our discussion with regards to metabolism. So this is the last unit that we will consider for our study of biochemistry. No? So who would have thought that it's as if it's the murag bago pa nagsugod ang semester, but currently we're now ending it. Anyway, so I've constantly mentioned this from time to time that the reason why we're attempting to understand the properties of the different biomolecules in the past is because we want to understand metabolism, the heart of biochemistry. So this is where we're going to understand what is really the essence why we're studying biochemistry. Because metabolism is already integrating what we've learned in the past and trying to get a clear picture of how they interact inside our body. And for me, metabolism, yeah, the study of metabolism can be confusing, can be intimidating, but when you understand its full concept, it's very, very much rewarding. Ang feeling ba, no? The feeling will be very, very rewarding. Kay aside from the fact that they're able to grasp a very complicated topic, but you will also realize ba nga kanang life is really marvelous down there at the molecular level ba nga. Grabe gud ang organization sa life, no? Sige. So let's now begin our study of metabolism, no? So even before taking up biochemistry, right? You've heard of metabolism already. Do you agree with me that metabolism has already been a word of mouth, even in our daily conversation? No, ako as a person, I'm usually given the comment nga paspas kaayo ang akong metabolism. So why would people tell me that I have a fast metabolism? What is their idea of metabolism di ay? Kaya nga ako manggong characteristic as a person class in terms of food, I eat a lot. Actually, when I go to Mang Inasal, I can actually finish up until 5 cups of rice or even 7 when I'm very stressed. But then again, I don't get fat. Diba? So that's why people will tell me that I have a very, very fast metabolism. No? But is it really right to tell me that I have a fast metabolism? No? What really is metabolism di ay? Correct? No? So, unsa mo gina metabolism then? Nganong ato man siya i-relate sa food immediately? Do you agree that when you hear the word metabolism, what comes to your mind immediately is the way we use food? Correct? No? So, what's really metabolism? So, that's what we're going to learn today. So, in general class, metabolism is just the sum total of all the chemical reactions that are going on inside the cell. Do you follow? Diba, we've talked about the central dogma of molecular genetics. You saw how the DNA is utilized to form the RNA, and then the RNA is utilized to form your proteins. Am I right? So kato siya, chemical reactions man to sila. So those are still also part of metabolism. Kay tanang mga chemical reactions that are going on inside your cell are part of the study of metabolism. In fact, metabolism can be divided into 2 types. It can either be catabolism or anabolism. So based on the illustration on your screen right now, how would you characterize, how would you differentiate catabolism from anabolism? Sige. Based on what you're seeing on your screen right now, there's a very, very good picture here that shows the difference between kata and ana. Yes, Christina.
I believe, sir, that catabolism is how it processes larger molecules to smaller molecules. Meanwhile, in anabolism, it clumps them together to form larger molecules.
Okay, that's it. No, that's it. That's exactly what it is. Yes, Christian, you would like to add something?
We could also say, sir, nga, catabolism releases energy and then anabolism requires energy.
Okay, very good. That's also true. So both of their answers are correct, and that sums up what catabolism and anabolism is. Okay, when you say catabolism class, you have a large molecule. For example, you have a biomolecule such as your carbohydrates, proteins, no? So when you break them down, of course, that's catabolism. But from the building blocks, nakita ninyo no nga, gikan kag monosaccharide, pwede man siya ma-converted into polysaccharide. Gikan kag fatty acid, pwede man siya ma-converted to form your lipids. Gikan kag amino acids, pwede man siya ma-converted to form your protein. So those processes that allows for the formation of larger molecules, that's anabolism. And in terms of energy, Christian is also correct. Ano man, large degradation of large molecules involves the breakdown of chemical bonds. Do you follow? Kaya kung naka large molecule, you want to make it smaller, you need to break down the chemical bonds between those molecules. The chemical bonds in your biomolecules class are very rich in potential energy. Therefore, when you break them, there will be a release of energy. Do you follow? So on the other hand, anabolism requires energy because in anabolism you're going to create chemical bonds man, no? So the creation of chemical bonds requires energy. 'Di ba? Kay mao man na siya kay 'di ba naman gitawag nga law of conservation of energy? No, remember mo ana sa inyong high school, no nga? Energy is not created nor destroyed. Correct? So kung pila to ang energy nga required to create the chemical bonds, mao sa tong energy nga iyang i-release. Do you follow? So that's the cycle, no? That's and kung imo na siyang i-sum up tanan, those chemical reactions, regardless if it's degradation or its formation, regardless, no? So that's what you called now, no? That's what you called now metabolism. So sum up ana nilang tanan. So what's the correct term for me? Ko ako nga, I eat a lot but I don't get fat. Is it correct that I have a fast metabolism? What should be the correct statement that should be used to describe myself? I have a fast. What do you think? I have a fast catabolism. Okay. Mauna nga, kung bang ingon gali ko nga, ala sir, kapaspas ni mog metabolism. What part? What part of my metabolism is fast? Di ba? See, it's catabolism. Okay? But there are actually processes class that falls in between. So we call them amphibolic pathways. So I will explain later the significance of amphibolic pathways. Okay? Sige. So mauna siya ang imuhang metabolism. So chemical reactions rana that are occurring inside your cell. Okay? Sige. So nothing so special about metabolism actually. Okay? Sige. So, and now, di ba? Okay, if you're going to look at it, no? Kata is degradation. Ana is synthesis. So there's a conflict between them. No? There's a conflict between them. No? However, class, in the cell, they do not occur actually in a manner nga kanang separated sila. They actually occur simultaneously. That's the irony of it ba? Nga mo degrade ang ato ang cell at the same time mo build up sad siya. So in other words, there's a conflicting demand between anabolism and catabolism. So how do our cells manage these conflicting demands? It is through compartmentalization, no? We call it what we call compartmentalization. So the different reactions that are involved in catabolism and anabolism are compartmentalized in different organelles of the cell. Maunang importante kaayo na ang ato ang mga organelles because in different organelles, there are different chemical reactions that are taking place there. For example, in the cytoplasm, naa mga to ang pag-synthesize sa imuhang lipids. Correct? Then kung adto po ka sa mitochondria yun, kaya adto man sa ang degradation sa imuhang lipids. Do you follow? So there's, although conflicting, no, kay ang usa kay nag-synthesize, ang usa kay nag-breakdown. Although they have conflicting, but because they are located in separate areas, they are compartmentalized, no? So that is how our cells manage this conflicting demands. Okay? So that's why nga, in studying class, no? In studying metabolism, we need to have a review about your cell, good, no? We need to review about your cell. Okay? Sige. So another important characteristic of metabolic reactions is this. When we write chemical reactions in the past, how do we write chemical reactions in the past? Diba? It's very simple, right? We have reactant, and then we end up product. Diba? This is how we write chemical reactions in the past. We have a reactant, we have a product. But when you say metabolism, for example, a very important reaction in metabolism is the conversion of glucose. No, this is a very important reaction in metabolism. The conversion of glucose to carbon dioxide and water. So in general chemistry, we just directly write that this way. And it reacts with glucose and oxygen, forming carbon dioxide and water. But in reality, this does not occur in the cell in a single step. So metabolic reactions occurs in terms of what we call pathways. So meaning to say, the reactant will undergo series of chemical reaction until reaching a certain product. No? So this actually allows our cells to to manage the energy ba. Okay, for example, class, kanimo gud imuhang kuan, kanimo gud imuhang oxidation sa glucose. This will release a lot of energy. So could you imagine if single step siya? So automatically, ikakaon mo glucose, mabuto ka. No, kay daghan mo kay energy no kung single step ra ba. Kung inana siya kadali buhaton ang chemical reaction. Am I right? You will explode because this will release a lot of energy man per molecule glucose. Am I right? But because it undergoes gradually in a pathway, it is able to manage the energy release. Nga dili ta makuyawan. Do you follow? Dili makuyawan atong lawas nga makamanage ta sa sudden energy surge. Okay? And in terms of pathways, there are different pathways, no? Na atay gitawag na linear pathway, cyclic pathway, and branch pathway. How would you characterize a linear pathway, class? How would you characterize a linear pathway based on the illustration there?
Hello, sir.
Yes, Benedict.
For me, sir, the linear pathway is just only a one-way reaction which has a final product.
Okay, very good. One way. I mean to say, no turning back, correct? That's actually correct. So you have a reactant in the beginning and you convert it to a product, but in multi-step reactions. Okay, very good. It's linear. So how about if it's cyclic pathway? For example, you have an initial reactant which is F. What do you observe in a cyclic pathway? Was it consumed? Was the initial reactant really converted into a completely different product or not? Was it regenerated?
I think, sir, it's generated.
Okay. In a cyclic pathway, it's regenerated. No? It's regenerated. Okay? So, sa linear mong good kay, naka-initial reactant, makonverted good siya at the end of product. Do you follow? But if it is a cyclic pathway, you have an initial reactant and then at the end of the cycle, it's regenerated back. Okay? So there's also an importance of the cyclic pathway later on. Okay? And then you also have a branched pathway. So if it's a branch pathway, so you have a reactant that can proceed in different directions. So that's a branch pathway. But then again, as you could see, linear gihapon na siya, pero lahi-lahi lang, there are a lot of possibilities. Okay? So that's how you describe metabolic pathways. So now let's consider the chemical reactions occurring in different parts of the cell. Like what I've mentioned, this is a very important property of cellular reactions, that they do not occur in the same place at the same time. Do you follow? I mean, yeah, they occur simultaneously, but in different areas of the cell. They are compartmentalized. So as you could see there, I think this is also written in your notes. So you would actually see there the different organelles, and listed there as well in the organelles is your different chemical reactions, no? Different chemical reaction or different metabolic reactions per organelle. Is that clear? So mauna siya. So I don't really want you to memorize them all nga peroxisomes kay mo na yung mahitabo, no? So it's very actually idealistic to let you memorize that, and at the same time knowing that not all of these processes can actually be discussed today, right? Kay gamay na lang tag time. So disclaimer lang daan ha, there's plenty of metabolic reactions. But for our class, we will only focus in metabolic reactions that are involved in what we call biochemical energy production. Is that clear? So again, in our context of the study of metabolism, we will only focus on the metabolic reactions that are involved in, that are involved in biochemical energy production. I mean to say, how are we able to produce energy? Kaya dili naman tarang tanan class kay murag it would take us a semester to finish everything. Okay? So that's why we'll focus on biochemical energy production. And speaking of biochemical energy production, the key organelle there, what do you think is the key organelle in biochemical energy production? What do you think is a key organelle in the biochemical energy production? Okay, your mitochondrion. That's why we call it the powerhouse of the cell, because most of the chemical reactions that are occurring in the biochemical energy production is occurring in the mitochondrion. Sige, so let's discuss in detail the anatomy of your mitochondrion. So mitochondrion, how would you characterize the shape of mitochondrion? It's shaped like a longanisa, no, like sausage. Am I right? No, croissant. Croissant. How do you, I don't know how the French pronounce that. It's croissant. It's croissant, no? In English, it's croissant, but it's shaped like a croissant. And then, what did you notice? When you cut mitochondrion into half, no? You would notice that it comprises two membranes. Do you follow? Di ba kaya itong cell kay natay cell membrane. So mao man din tong membrane sa tong cell. Am I right? But kani mong mitochondrion kay funny kay na siya kay ikakati mo na siya membrane sa gawas, and then inside pag na po na siya ikaduhang membrane. So there's an outer membrane and an inner membrane. And then the inner membrane, how is it arranged inside the mitochondrion? It's actually no? So you have an outer membrane, and then the inner membrane is actually arranged in curlicues, no? Like curlicues inside your outer membrane. Kanisha, this is your outer membrane, and this is your inner membrane. So how do you call this area here? How do you call this area inside, no? Inside the inner membrane. You call it that's your matrix. Okay, always remember you need to be very familiar about the anatomy of mitochondrion. Okay, mag-focus man tag biochemical energy production and dili man na sila mo occur. Di follow? Okay, para makaibaw mo ba na mo idea, maka-picture out mo ay okay, ari din the pita sa mitochondrion, no? So it's in the matrix, no? Inside the inner membrane is the matrix. And then how do you call the space between the inner and outer membrane? So the space between the outer and inner membrane, you call it the intermembrane space. That's your intermembrane space. And for every curve of your koan, for every curve of the inner membrane, that's your crista, okay? Or the plural form cristae, no? If it's a lot, it's cristae. But if it's single, then it's crista. Do you follow? No, so kanang curves imong mitochondria. Okay, so that's it, no? That's the anatomy of your mitochondrion. So later on, when I tell you that this reaction, of course, in the matrix, okay, ilan na mo asa dapit sa matrix. Kung maingon ko nga, this reaction, of course, in the intermembrane space. So you know already where is the intermembrane space. Okay, so that's your mitochondrion. It's a sausage-shaped organelle, no? So yeah, so I wasn't able to save the picture class, but we had an experiment before in our biochemistry where we need to look into a mitochondrion. Okay, in our school we have a scanning electron microscope. So that is what you're going to use to look at into the different organelles. So not to brag, but I was the only kind of student who was able to prepare a mitochondrion slide. Okay, dili mo guna siya sayo, no? It's not very easy actually to make a slide ba, no? Kuan sa mga organelle. Gagmay na gud kaayo na class, di ba? So yeah. Anyway, so this is really what it looked like, no? Inani siya dagway, siya sausage shape, and then naa siya mga membrane sa sulod. Okay? Sige. Question so far? Question so far? None? All right, so let's proceed. So now let's proceed to the key metabolic intermediates. Okay, again, unsa gay itong mga part sa metabolism ra ang atong i-consider for biochem nato? We will only consider those that are involved in biochemical energy production. So therefore, kani nga mga substances nga ato ang hisgutan, these are also the substances that are involved in the production of energy. Okay? Sige. So when we say intermediates, these are not product. Do you follow? Always remember, these are not products of the reaction. Instead, these are normally byproducts. These are helpers of the chemical reaction. Do you follow? Okay. So what are these helpers and what are their function in metabolism? The first intermediate is what we know as your adenosine phosphate. And you know, you are already familiar with this. Am I right? We already introduced to you before your adenosine phosphate in our study of, asa man nila to na ilas in adenosine phosphate class, in the study of your nucleic acid, correct? So how would you characterize adenosine phosphate? Is it a nucleic acid, a nucleotide, or a nucleoside? An adenosine
Nucleotide, sir.
a nucleotide because it comprises a nitrogenous base, a pentose sugar, and a phosphate group. So for adenosine phosphate class, no? From the name itself, adenosine, no? So what nucleotide is, what nitrogenous base is present? It comprises adenine. And then what sugar is present? Ribose or deoxyribose? Ribose.
Ribose.
Okay, so maon na siya, no? And then your adenosine phosphate can accommodate up until 3 phosphate. That's why we have 3 types of adenosine phosphates. We have AMP, which stands for adenosine monophosphate, and we also have ADP, which stands for adenosine diphosphate, and ATP, which we're very already familiar, right? Adenosine triphosphate. This is known as the energy currency of the cell. So remember, no? So remember, during catabolism, sa ito mahitabo, mo release og energy ang chemical reaction. So asa man to ang energy karon ika-release sa catabolism, ipanglabay ra nato no sa ato rang i-release environment. O so dili. So the energy that is released in catabolism will then be stored in your ATP. So if you're going to ask me, sir, among all the 3 adenosine phosphate, why is it only ATP that is actually considered to be an energy currency? Okay, so it has something to do with the number of phosphate groups. Okay, kaya nga naman class. Okay, so let me describe to you the structure of adenosine phosphate. When a phosphate is directly attached to a sugar, kani ba kani nga bond, no? This is the attachment of phosphate directly to the sugar. That bond is considered an ester. Do you follow? Do you follow? A phosphate attached directly to a sugar, the bond connecting them is an ester bond. Karon, what if there's another phosphate attaching to another phosphate? So kana. So these are already what you call your anhydride bonds. Okay, so when a phosphate and a phosphate is attached together, that is no longer an ester bond. Instead, we call it an anhydride bond. So in terms of energetics, an anhydride bond has more energy than ester. So kinsa may pinakadaghang anhydride bond? AMP, ADP, or ATP? It's your ATP, right? Because it contains 3 phosphate. So because of that, that's why nga, it's an efficient storage form of energy. That's why nga, it's the energy currency of the cell. Because it comprises 2 anhydride bonds that are highly energetic. Do you follow? No? Do you follow? Okay. So mauna nga, ang atong pag-store sa energy kay in the form of your adenosine triphosphate. Okay? Yeah, yes, nalagyan po energy, but lesser siya, dili kaya siya ginagamit nga storage kay ang concept sa atong cell, kung asa siya maka-maximize, adto siya. Di ba? Asa man siya maka-maximize og store? Adto sa ATP. Okay. Although maka-store og energy sa ADP o sa AMP, but to a very minimal amount. So, naagya po yung excess nga dili ma-store. So, adto siya nga mas efficient ang storage. No? So, our cells are very intelligent that way. Okay? Kay bawal siya. Okay? So, again, adenosine phosphate. Unsa sila gamit? What's the use of your adenosine phosphate? They are used for energy storage. Okay? Sige. So next, no? So next, intermediate. So what are our next intermediates? So now we have your, so the next two intermediates class, no, we have two next, no? So we have your flavine adenine dinucleotide and nicotinamide adenine dinucleotide. So the next two are actually involved in what we call oxidation reactions. Do you follow? So the next metabolites that we will be discussing are coenzyme. No, akong specify. These are coenzymes. No, coenzymes of oxidation reaction. So meaning to say, if ang imuhang metabolic reactions can nag-involve of oxidation, you will expect to see a FAD or a NAD that is utilized. Do you follow? No? So kung naay chemical reaction sa cell ba nga nag-require of oxidation, dapat it has a concomitant use of FAD and NAD+. Okay? So let's discuss first your FAD. FAD stands for flavine adenine dinucleotide. From the name itself, it's the adenine dinucleotide. Ano man, yung itawag man siya adenine dinucleotide because it comprises an ADP, no? Adenosine diphosphate. One of its moiety, it's an adenosine diphosphate. So the adenosine diphosphate is further attached to what we call a riboflavin, no? So basically, a flavine adenine dinucleotide is just an ADP attached to a riboflavin. So riboflavin is actually comprises a ribitol and this very tricyclic ring here. This is called a flavine, no? So riboflavin class is basically your vitamin B2. Do you follow? It's just your vitamin B2. So in other words, flavine adenine dinucleotide is an ADP attached to vitamin B2. So remember when you were in elementary or perhaps in high school, it was always mentioned that vitamin B are used for metabolism. Am I right? But we haven't really known sa yung gamit sa metabolism. Oh, this is one use of the vitamin B. It is utilized in coenzymes, in the intermediates of metabolism. Do you follow? Do you follow? Okay? So unsa may purpose aning flavine adenine dinucleotide class? It's an electron acceptor. Okay, always remember, during oxidation, when a compound undergoes oxidation, it loses hydrogen and it also loses electrons. Always remember, huh? When a compound undergoes oxidation, it loses hydrogen and at the same time, it loses as well electrons. So the hydrogen and the electron that is lost by a compound as it undergoes oxidation will be gained by your flavine adenine dinucleotide. So maunang gitawag nato siya nga electron acceptor or electron transporter. Do you follow? Do you follow? No, so kani, no? So always remember class, for every oxidation reaction, there is always, no, so when a compound loses, when a compound undergoes oxidation, there's always 2 hydrogen and 2 electron that is lost. So ang katong hydrogen o electron nga na lost sa compound, mag-gain to siya sa imuhang FAD. So that is why nga from FAD, it was converted into its reduced form, FADH2. So the H2 there is to indicate that it is able to accept 2 hydrogen atoms. At the same time, aside from 2 hydrogen, timan isad gyud nga naka-accept sad siya og 2 electrons. Do you follow? Aside from hydrogens, it was also able to accept 2 electrons. Am I right? Am I right? Okay, kasabot rata anak class. So class, kana dako kay ang flavine adenine dinucleotide, no? But this is only the operative part. Kanira siya ang gamit. Nga naman, because that is where the electrons and the hydrogens are gained. Do you follow? No? So why gamit ang uban? Kung ano siya? Pakapin sa abubot, no? Wala siya kuan ba? Wala siya sa kanang kuan, no? Wala siya kanang kuan lang. Walang. Para na ma-complicated atong life, no? Kaya ganahan lang yung si biochem nga complicated, no? But this is only the operative part, not only your flavine, because that is where the oxidation reaction will take place. Okay? Sige. So by the way, so always remember, if the oxidation is brought about the removal, no? If the oxidation is the conversion of single bond to double bond, then that is where your FAD is used. Timanin na ha? If the chemical reactions involves the conversion of single bond to double bond, then that will utilize your FAD. Okay? Sige. Question so far? Question so far? Wala ra? Okay, so let's now proceed to the next metabolite. We have your NAD+. Okay, ano mang NAD+? So NAD+ stands for nicotinamide adenine dinucleotide. So just like your FAD, it's also comprising a adenosine diphosphate. So that's why it has an adenine dinucleotide. But however, so may difference niya sa FAD. Instead of a riboflavin, your ADP is further attached to a ribose. And then the ribose is attached to this group here. This group is called a, kaniba, kaning green nga portion. That's called a nicotinamide. So where do we obtain nicotinamide? We obtain this from niacin. You know what niacin is? Niacin is vitamin B1. Do you follow? B32? What's it? Vitamin B32, Ben? B3 ba? I think it's B1. Try to check. I think it's B1. B3, no? B3 class? Yeah, vitamin B3. Yeah, it's vitamin B3 because B1 is biotin, no? B1 is biotin. B3 is, yeah, right? It's niacin. Okay? So it's vitamin B3. That's correct. Okay? Sige. So that's your nicotinamide adenine dinucleotide. So again, no? So nakita ninyo nga vitamin B gihapon ang useful class. Am I right? Magamit gihapon si vitamin B. All right? So nganong naman siya, nganong ang iyang abbreviation kay NAD+ man? Because if you try to look at here at this portion of your nicotinamide, it has a positive charge, no? Ang imong nicotinamide portion baka na positive charge. That's why nga NAD+. Do you follow? Do you follow? Okay. So the same na ang function ni NAD ug ni FAD class, di ba? During oxidation kay ang compound mo lost og hydrogen og electrons. So ang gamit sa dinad, iyan sa nang i-accept, just like your FAD. It will also accept the hydrogens and the electrons. But what's the difference between NAD and FAD? Kang FAD kaganiha class, you notice that, tawa, both the hydrogens and the electrons are all gained. Do you follow? The 2 hydrogens and the 2 electrons are gained by FAD. But in the case of your NAD, what did you notice? Only the 2 electron is fully gained, but only 1 hydrogen is gained. No? Among the 2 hydrogen that is lost during oxidation, only 1 hydrogen is gained by NAD. So that is why nga, after the reaction, you still get an H+ nga excess. That's why nga, ang reduced form ni NAD, kaya ato siyang gitawag na NADH. Do you follow? We call it NADH to indicate that it only received 1 hydrogen. Do you follow? But the 2 electrons are fully gained, okay? So no question about the electrons. It's just that it is only able to accept 1 hydrogen ion. Okay? So si FAD kay, di ba si unsa itong reaction si FAD? FAD if it's single bond to double bond. Am I right? Si NAD, unsa man si NAD? So NAD is between alcohol to carbonyl. So if there is a reaction that involves the conversion of alcohol to a carbonyl, then that requires your nicotinamide adenine dinucleotide. Do you follow? So you will be able to produce a NADH. All right? Is that clear? Is that clear? Okay? Am I making sense? Class? Again. So for example, taganda mo example ha, kay para dyan mas masabtan ba, no? Kay kung walay example mo, sige na tag-ingon, kay ang mga estudyante kay dili sa ka-remember mo, hindi nga, wa man nagipagawa sa lesson, sir. Alam no, lain sige nga, tanang nga akong i-exam kay pagawa sa lesson. Dili ko ma-exam, no? Manghatag lang kung answer key, no? Kung ipagawas mo i-lesson mo nila ko tanang i-exam, di ba? Sige. What do you think is the coenzyme that will be utilized for this reaction? Okay. What do you think is the What do you think is the coenzyme that can be used for this reaction? Will we use NAD or FAD? So we will use your FAD. Okay. So initially you react this with FAD, right? You want a FAD, right? So after the reaction, what happens? Your FAD is converted to what? It's converted to FAD, and then how many hydrogen? FAD H2. Am I right? Am I right? No? Sige. So, okay. Inanaon na siya pag-analyze, ha? Kay basig ma-blame ko ba nga sa exam mo gawas manilin. Wa man nag-mention, o, di ba? Anak, gid ba na mo estudyante kay dili mo think outside the box ba, no? Sige. Anyway, so let's continue. Walay question about NAD and FAD? So far, so good. All right. So the last metabolite that we will be talking is your coenzyme A. So this is number 4. Anyway, anong 3 pa maghihapon na? Okay? So this is supposed to be number 4 already. Baka mislead, no? The 4th one is your coenzyme A. So this is the largest class among all the metabolites we've talked earlier. This is the largest. So what are the different parts in your coenzyme A? First, of course, it contains an adenine, no? Bida, kasi adenine plus, no? Bidang bida, no? So you have an adenine, and then it's attached to a phosphorylated ribose. What do we mean a phosphorylated ribose? It's just a ribose, but at carbon number 3, it's attached with a phosphate group. Do you follow? A phosphorylated ribose is just a ribose, but carbon number 3 is attached to a phosphate. And then, attached mo gid siya og another phosphate group, and then attached sa ani pantothenic acid. So pantothenic acid is your vitamin B5. O, diba? So vitamin B na sun, no? Grabe yung kasikat sa si vitamin B. So it's attached to vitamin B5, and then the pantothenic acid is further attached with what we call an amino ethane thiol. Okay? So kasakadako sa atong molecule class, this is the operative part. Could you imagine, no? Dako kay siya, pero wala, again, pakapin sa abubot ang uban. No, why gamit, no? So only the amino ethane thiol that is very, very important. So what is the function now of coenzyme A? It carries acetyl. Acetyl class, ha? Ang acetyl ba, it look like this, no? An acetyl molecule looks like this, CH3, C double bond O. So this is an acetyl. So kanis siya, this is a very important group. Later on, maka-realize from later on sa importance ana. But I want you to know from now on, no, that the acetyl group is very, very important in metabolism. So that acetyl will be carried all throughout the cell by the action of your coenzyme A. So once the acetyl will attach to the amino 18 thiol, it will form what we call a acetyl coenzyme A. So that's why class nga ang kanang coenzyme A, it's normally it's normally abbreviated this way. Okay, imagine no mag-drawing gid ka sa entire acetyl coenzyme A. Doing mo gini mo appeal, no, so wala. No, so that's why nga it's often abbreviated this way. So mean to say it's an acetyl attached to the thiol group of my coenzyme A. Is that clear? Is that clear? Okay, so that's your, so once it's already attached with acetyl, it becomes acetyl coenzyme A. All right. Okay, so these are now the four different metabolites. Okay, so we have adenosine phosphate, which is used to store energy. We have the FAD and the NAD involved in oxidation reactions. And then lastly, we have your coenzyme A, which is utilized as carrier of acetyl group. And there's already the significance of acetyl group later on. Do you follow? Do you follow? Sige. Questions, ladies and gentlemen, from the Republic of the Philippines? Questions?
None, sir.
Okay, none. None for, okay. So before I'll proceed to the next topic, I will be giving you 5 minutes break in order for you to relax, okay? So let's be back at 2:54. Okay, let's be back at 2:54. Is that clear? Sige. So before our break, we already talked about the overview of metabolism. No, so we're already aware of what metabolism really entails. So kahibaw na tanga, when we talk about metabolism, it is simply the sum total of what is going on inside your cell right now. No? So as you live and breathe, you are doing metabolism right now in your cell. And it can be comprising anabolic or catabolic reactions. So this diagram here, I found this in the internet. This is a very, very beautiful diagram showing the interrelationship among the different metabolic pathways. Diba? I've mentioned to you that metabolism can be studied in terms of pathways. So kaning mga gluconeogenesis, mga glycolysis, these are what we call the metabolic pathways. And always remember ha, kung moingon tag metabolic pathways class no kay, always remember it means that that's actually multi-step. So when we say glycolysis, it does not occur in one step only. It occurs in multi-step. So when we say gluconeogenesis, then that also not occurring in one step as well. Okay? So kana sila summary na ni siya, no? So these are not the entirety of metabolic pathways class, but these are just the metabolic pathways that are involved in, specifically, I would just like to emphasize, no, kay basig mamislead mo ba? These are the metabolic pathways involved in biochemical energy production still, okay? So these are the pathways involved in the production of energy, okay? So in this diagram, to help you interpret, anything going down is catabolic. Do you follow? So anything going down in the pathway in the diagram is catabolic, and anything going up, ang kanang arrow going up is anabolic. Therefore, for example, how would you characterize gluconeogenesis? Is it anabolic or catabolic? Based on the diagram. It's basically, asa man yung arrow, but it's actually going up. So therefore, it's anabolic, no? So the conversion of pyruvate to glucose, it's anabolic, okay? But basically, as I pasabot gani kung anabolic, it's build up from small molecule to a larger molecule, because pyruvate class is a very small molecule. And as we know, Glucose is already a large molecule. Therefore, that involves anabolism, sorry. So how about beta oxidation? So how would you characterize beta oxidation? Is it kata or ana? This basic, yung pagkata-ana, dapat i-full ninyo. Is it catabolic or anabolic? Okay. Basig maanad mo kata o ana ba? Sige. So based on the diagram, how would you characterize beta oxidation pathway? Is it catabolism or anabolism? It's, okay, fatty acids converted to acetyl coenzyme A. That's spiraling down, catabolism, no? Catabolism, because acetyl coenzyme A is a very, very simple molecule. Okay? So what did you notice in this diagram, class? Diba? You started with different macromolecules. You started with lipids, carbohydrates, and proteins. But what did you notice? Eventually, they will all lead up to a common byproduct. And what is their common byproduct? They will all lead up, no, regardless, for example, nikaon ko lipids, proteins, or carbohydrates sa lunch. No, so very different biomolecules. But where are all they leading up to? No? ATP na yun? Asa mo sila mo converge first? They converge in what byproduct? Do they first converge? Makita, kang hala silang tanan pa, ingon gud to. So, they all converge in acetyl coenzyme A. So, maunang giingnan ta mo nga, the acetyl is a very important group because our body would tend to degrade the food that we eat into acetyl. So, katong acetyl, mo attach na dayon to siya sa atong acetyl coenzyme, sa atong coenzyme A, forming the acetyl-CoA. So, the acetyl-CoA will then undergo the Krebs cycle and the electron transport chain. So this portion in metabolism is known as the common metabolic pathway. Okay? So this area here in metabolism is known as the common metabolic pathway that comprises your Krebs cycle and your electron transport chain. Why do you think it's actually called a common metabolic pathway? Because regardless if you are actually coming from, protein, lipids, or carbohydrates, you will end up to that pathway. Diba? So nindot kayo, no? I-realize mga different kayo mga biomolecules, but in the end, in the cell, they will all end up in the same pathway we call the common metabolic pathway. Okay? Sige. So that's it, no? So yeah, I don't intend you to memorize that, no? So I'm just showing this to you to give you an appreciation that is what really how it happens inside your cell. And that's not just it, ha? Gamay ra kani siyang portion, class. Mas naapog mas daghan ana. Okay? Sige. All right? So let's now proceed to the main topic in our understanding of biochemistry. Like what I mentioned, we want in our study of metabolism, we want to understand biochemical energy production. Okay, so let's begin discussing about biochemical energy production right now. So from what I've mentioned, biochemical energy production is the process that allows our cells to obtain the energy stored in the food that we eat converted into usable form of energy. Specifically, that usable form of energy that I'm talking about is their adenosine triphosphate. So biochemical energy production comprises 4 stages. Stage 1, stage 2, stage 3, and stage 4. So in stage 1, you see there, so this area here is stage 1. What did you notice? What did you notice in stage? Wala pala ginagkita sa akong screen ang akong gi-encircle, no? So hello? So kani siya nga area diri class ba, no? Unstable lagi akong internet sa kuan. Anyway, so this area here, can you see what I'm encircling? Can you see it? The line I'm encircling?
Yes, sir.
Okay, so that area there comprises stage 1. So what happens in the stage 1 of your common metabolic pathway? What happens in stage 1 of the common metabolic pathway, class? Did you notice?
Breakdown, sir, of the biomolecules into their smaller units.
Smaller units into their corresponding building
Monomers.
blocks or monomers. Very good. Okay, so this is stage 1 of the biochemical energy production, correct? No? So again, in the stage 1 of biochemical energy production, of course, you're going to first degrade them into monomers. Kay dili man na makasulod ang proteins, carbohydrates, or fats directly inside the cell, class. Only their building blocks can directly enter the cell. And remember, biochemical energy production occurs inside the cell. So in order for them to enter the cell, they need to be first degraded into their specific monomers or their specific building block. So the other name of stage 1 is, unsa may other name? Unsa man nga process sa atong body ang mag-convert sa mga macromolecules into their building blocks? That's called
Digestion.
digestion. Very good. It occurs in the what system? In the digestive system. So what are comprising in the digestive system, class? You have your mouth, followed by diretso stomach imo dear. Para sa una lang no. Alien, alien no, diretso way esophagus no. So mo esophagus sa followed by your stomach and then your intestine, no? The intestines are comprises the small and the large. Kinsa may maguna, small or large? Kasi maguna, small or large intestine?
Small.
Small. Then the last one is your rectum. Okay? So where does ingestion occur? Ingestion occurs in the mouth. This is where ingestion. And then where does defecation occur? Defecation occurs in rectum. Okay? Where will absorption occur? Absorption is in the small intestine, no? Small intestine specifically. Do you follow? Absorption occurs in the small intestine. So it cannot occur in the opposite direction, class ha? Dili pwedeng ang ingestion kay sa rectum magsugod. Kuyaw, no? So dili na pwede. So dapat ingestion should always begin in the mouth. No? Wala mo siguro pagkaon, no? Nga nagsugod sa rectum. Do you follow? Okay? So the purpose really of your digestion is to convert the macromolecules into their building blocks. And these building blocks or monomers will then be absorbed in the small intestines to be transported into the different parts of the cell where they are needed. Okay? Sige. So what is now stage 2 of your biochemical energy production? So nanakay mga monomers, okay? So naka-convert na ka sa imuhang ginatawag nga mga foods into their building blocks. Karon, imo na din to siya i-convert sa ilang common byproduct. Ano sa ilang common byproduct? What is their common byproduct? Again, their common byproduct is your acetyl. So in stage 2 class, you convert the building blocks into their corresponding acetyl coenzyme A. So always remember, class, lahi-lahi na silang kuan, lahi-lahi na silang way. Is that clear? So stage 1 and stage 2 is very different from the different biomolecules, no? Lahi ang digestion o formation ni acetyl ni carbohydrates. Lahi kang lipids o lahi kang proteins, no? So there are different ways to do that. So that's why nga they converge. So once they are already converted into acetyl coenzyme A, that's the time that they will undergo what we call the common metabolic pathway. Kaya regardless if asa na siya gikan nga acetyl-CoA, they will actually proceed to the same pathway. So the first entrance of the acetyl-CoA, they will enter in what we call the stage 3, which is your Krebs cycle, the citric acid cycle. Okay? So afterwards, after the citric acid cycle, no? So the acetyl will be oxidized there, converting them to carbon dioxide, and then afterwards, no, naaman yung mga byproducts ang imong Krebs cycle. So the Krebs cycle now, the byproducts of the Krebs cycle will then proceed to your electron transport chain, no? It will then proceed to your electron transport chain, and in the electron transport chain and the oxidative phosphorylation, the last stage, that is where your ATP is produced. Okay, for our one class. Okay, so again, no, so that's what happened, no? So again, so your sugar, I mean, no? So that's it, no? So after the conversion of acetyl coenzyme A, they will then undergo your common metabolic pathway. Okay, so for today, we will now consider the different common metabolic pathways. For us to understand first, asa mo gini sila paingon tanan, di ba? So let's consider first the common metabolic pathway. So the next topic supposedly Wednesday, kay kato na untang stage 1 o stage 2 ni carbohydrates, proteins, o lipids. But then again, since we cannot meet on Wednesday, so I'll just give it to you as a recorded lecture. So for today, we'll just finish the stage 3 and stage 4. Anyway, it is common to all of our biomolecules. Okay? Sige. So let us begin discussing about your Krebs cycle, or also known as a citric acid cycle, or in other textbook, it's known as a tricarboxylic acid cycle. Okay? So this is a very infamous cycle. And you know what, class, no? So, kanira gid ang ako ang hangyo ninyo, no? Diba? Daghan man kayo nig pathways. There's plenty of pathways here. And you know, I don't want you to memorize the other pathways, but please promise me that you will memorize the citric acid cycle. Is that clear? Kana ara siya. You memorize the citric acid cycle. Is that clear? Is that clear? Among all the other pathways, bahalag dito niyo i-memorize. Basta i-memorize niyo ang citric acid cycle. Okay, sorry class kayo. Nag-share mo kung office karon so medyo kuan daghang tao diri. Anyways, again, like what I've mentioned, when we talk about the different pathways, kanira ang akong ihangyo no nga, you memorize the Krebs cycle. Anyway, it's just good for 8 steps naman siya. So, ayon mo ka hadlok aning Krebs cycle. It looks very intimidating, but it's really friendly and it's actually very important to us, okay? So, in order for us to understand, kay para dali ra nato ma-memorize si Krebs ba, we need to really understand its components and its reactions. In order for us to know, no? In order for us to have a clear idea as to what is really happening there. Again, asa gini nilapit si Krebs cycle, it is the stage 3 of biochemical energy production. Do you follow? It's a stage 3 of biochemical energy production. So this is where your common byproduct, which is your acetyl-CoA, enters the common metabolic pathway. Okay? Sige. So the citric acid cycle is comprised an 8-step cyclic pathway. So you have a initial reactant and it's regenerated back at the end of the cycle. Okay? Sige. So don't worry, we'll go to these different steps one by one. So ang goal of the citric acid cycle class is very, very simple. It wants to oxidize acetyl. Diba kaniba? Diba? Ang imong acetyl-CoA, kaya na siya acetyl nga naka-attach. So the acetyl group of your acetyl-CoA, it will be converted to 2 carbon dioxide. That's just the goal of your citric acid cycle. So maunang dili siya lisod. It's just that, again, like what I've mentioned, metabolism occurs in pathways, step-by-step process. Do you follow? So the goal there So the goal there is really just to convert your acetyl to 2 carbon dioxide. Okay? Sige. So let's go. So what did you notice, class? Acetyl na ay duha ka carbon, gi-convert na to siya into duha ka carbon dioxide. How would you characterize that reaction? Carbon na himong carbon dioxide. There is a gain of oxygen. That's
Oxidation, sir.
oxidation. Very good. So therefore, ang atuang citric acid cycle would involve oxidation reaction just to convert the acetyl into 2 carbon dioxide. So unsa gay itong mga coenzymes, class, ang ginagamit sa oxidation process? The coenzymes, the 2 coenzymes are your NAD+ and FAD. Okay, so when they undergo oxidation, when they're involved in oxidation, they are converted to NADH and FADH2 respectively. So that's what we're seeing here. So after the Krebs cycle, because we're trying to oxidize your carbons in the acetyl to carbon dioxide, then we are actually producing as well concomitantly, mean to say as a result, no? We're also producing NADH and FADH2. Okay? So where is the Krebs cycle located? It's located in the matrix. So again, where is the matrix again? Where is the matrix located in the mitochondrion? It's located in the where? Where is the matrix located? It occurs in the inside of the inner membrane. Okay, so that is where your citric acid cycle is located. And then it's also considered an anaerobic process. What do we mean by anaerobic process? Meaning it does not require oxygen. Do you follow? It is a process that does not require that does not involve oxygen. Okay? So diba? Kaya ang kana ato ang kuan class, ang kana atong citric acid cycle, it's oxidative man. So you will expect to have reduced coenzymes NADH and FADH2. Unsa ganito ang gigain sa ato ang NADH and FADH2? What do they gain? They gain, so after the reaction ba, no? So na oxidation. So after oxidation, unsa man ang nag-gain sa imuhang NADH and FADH2? They gain what? They gain hydrogen and electrons. Correct? So ang kaning mga di ba? So na siya electrons, no? So NADH si FADH2 kaya na electron. So muadto na din to siya sa imong gitawag nga electron transport chain. So in the electron transport chain, ladies and gentlemen, that is where you see that the electrons, no? That the electrons in your in your NADH and FADH2 are being transported, no? Into different proteins. No, ang kanang mga electron transport chain class, it comprises different proteins. So the electrons that is gained by NADH and FADH2 from the Krebs cycle will then be passed on to this chain of proteins. So upon the transport of electrons in this chain of proteins, no, it will be able to gain energy. So the energy that is gained by electron transport is used to fuel what we call oxidative phosphorylation. So in oxidative phosphorylation, this is where you see the production of your ATP. Is that clear? Is that clear? No? So that's the coupling between Krebs cycle and electron transport chain. Do you get what I'm trying to emphasize here, class? Do you get what I'm trying to emphasize here? No, so again, because Krebs cycle, I mean the goal of Krebs cycle is to oxidize the acetyl of acetyl coenzyme A to carbon dioxide. So we'll produce reduced coenzymes nga NADH and FADH2. So si NADH and si FADH2, naaman siya mga electrons, diba? Nag-gain man siya electrons. So katong mga electrons nga ayan nag-gain, muadto to siya karon sa electron transport chain. Magpasa-pasa siya dito, siya nagpasa-pasa og bola ba, no? So ika-add to niya sa electron transport chain, the electron transport chain will be able to gain energy. So the energy will fuel the oxidative phosphorylation to produce your ATP. That's 10 seconds of biochemistry already. Do you follow? That's 10 seconds of the story of biochemistry. So if you get that concept, then you're good to go. No? You're good to go. That's really the use there. Is that clear? Am I making sense, everyone? Am I making sense? Class, am I making sense?
Yes, sir. Yes, sir.
Okay,
Yes,
very
sir.
well. Sige, so let's go now to the different reactions one by one. Okay, so this is where it gets messy because, you know, again, as a goal, we want to convert acetyl to carbon dioxide. Then again, it occurs in 8 steps. Okay, so kung 8 steps man, there are also 8 substrates, no? There are 8 substrates in your Krebs cycle. So of course, 8 steps, so there are 8 substrates. So what are the 8 substrates, no? So to help you memorize the 8 substrates, you can use the mnemonic on campus. I always kiss. Medyo green, no? Medyo kuan siya. Some pag yun, so daghan. Sexy, fine, men. Okay? You could use this mnemonic to memorize the substrate in the Krebs cycle. So on campus, I always kiss some sexy fine men. So if you're not comfortable with men, so probably you can use maiden. Okay? But what does it stands for? It stands for, so in order na siya class ha, in order as it occurs in the cycle. Beginning with oxaloacetate, followed by citrate, followed by isocitrate. Always KISS stands for alpha ketoglutarate, the 4th substrate, and then alpha ketoglutarate followed by succinyl coenzyme A, and then succinyl coenzyme A is converted to succinate, and succinate is converted to fumarate, and fumarate is converted to malate. And because it's a cycle, malate is regenerated back to oxaloacetate. No? So that's how you try to memorize the 8 substrate using that mnemonic. Okay, so there's plenty of mnemonics online actually that you can use, but this is something that I am fan of using because this was the one being taught to us by our teachers. Okay, so that Krebs cycle. Okay, sige. So let's go to the different reactions one by one. Okay, again, what's the first substrate of the Krebs cycle? The first substrate is your oxaloacetate. So what happens in oxaloacetate, no? So ang mahitabo class, it will combine first with your acetyl. Correct? So diba? Your acetyl-CoA carries acetyl. So the acetyl will now combine with your oxaloacetate. Okay, sige. Let's try to look at there. How many carbons does oxaloacetate have? It contains 4 carbons. Okay? How about your acetyl? Kaning nga portion, acetyl lang ha? How many carbons are there? There are 2 carbons. Do you follow? So, they will combine together. The acetyl will condense. No? So, this is a condensation reaction. So, it will combine together to form now your citrate. Correct? So how many carbons do you think? No, kaya dili man na pwede mausab ang number of carbons. So 4 plus 2, that becomes how many carbon? So citrate is already a compound containing 6 carbons. Do you follow? So that's the first step of the Krebs cycle. The acetyl will condense with oxaloacetate forming citrate. Do you follow? The enzyme responsible to that is known as your citrate synthase. Okay? It's known as your citrate synthase. Okay, so natangtang si acetyl kay it's removed from acetyl coenzyme A. So it combines now with oxaloacetate forming your citrate. Is that clear? Is that clear? No? Class? Am I making sense that in the first step? So of course, kay mo na siya technique sa atong cell bakay para easy ra niya i-oxidize later on si acetyl to carbon dioxide. Class, kasabot ra ta anak class, yes or no? Hello, are you still here? Hala, asa naman ako mga estudyante, no?
Yes, sir.
Yes, sir.
Yes, sir. Yes, sir.
Kaya maragwa na may ko'y nadunggan, no? Asa naman mo? Okay, sige. So that's the first step, okay? So always remember, class, the first step is a regulated step. Do you follow? What do we mean by regulated step? Meaning to say, it's like it acts as a faucet, no? It acts as a faucet, okay? So nganong faucet man? Because it can turn off the Krebs cycle. Okay, of course, the Krebs cycle produces a lot of byproducts. Always remember, huh? Krebs cycle produces a lot of byproducts. Sometimes the byproducts will become in excess, meaning to say our cell will no longer need these byproducts. For that reason, it needs to be turned off. Just like when you're having a faucet ba, when you actually open the faucet to get water, kung mapuno na ang imong balde, you need to turn it off. So one of the faucet in the Krebs cycle is your step 1. Because if you are already producing a lot of NADH class, no? When you produce a lot of NADH in the body, okay, you know, Krebs cycle produces NADH man. So when you already produce a lot of NADH, it will signal step 1 to stop. So when step 1 stops, the entire cycle stops. Kaya dili man siya makaproceed. Am I making sense? Am I making sense? So it's like a faucet in the Krebs cycle. Okay? Do you follow? Do you follow? Hello?
Yes, sir. Yes, sir.
Okay. Sige. So that's your step 1. Converting oxaloacetate, so combines with acetyl, forming now your citrate. Sige. So how about your step 2? Sige. In step 2, this is classified as a isomerization reaction. Mean to say, it's just rearrangement of atoms. So specifically, what is rearranged is this OH. Tawa. No? The OH change its position. So that's just your step 2. From citrate, it was converted to isocitrate. Do you follow? But what is the difference between citrate and isocitrate? The location of the alcohol group. Because your citrate contains an alcohol. So in citrate class, how would you characterize this alcohol? Is it primary, secondary, or tertiary?
Tertiary.
That's tertiary, correct? No, that's tertiary. How about in isocitrate? After the isomerization reaction in step 2, what happens to the alcohol? It was converted into primary, secondary, or does it remain as tertiary? It was converted to what type of alcohol?
Secondary.
Secondary. Okay, so anong kailangan may convert into secondary? Remember, Krebs cycle involves oxidation. Karon, ang kanatong tertiary alcohol, it cannot be oxidized. It cannot undergo oxidation. Therefore, bright sa natong cell, no? So because it cannot undergo oxidation, sige, it converts sa sitika into a substrate that can be oxidized. That's why it was converted first into isocitrate, because the isocitrate now contains a secondary alcohol, which can already undergo oxidation. Is that clear? And the enzyme capable of doing that is your aconitase. Aconitase. So that's the second step of the reaction, converting citrate to isocitrate. Significance of this reaction kay para ma-oxidize further ang atoang substrate. Okay? Sige. So ready na no? Ready na for oxidation. Pag converted na niyan to isocitrate, it's now ready for oxidation. So therefore, the third step is already the first oxidative reaction. So the third step is known as oxidative decarboxylation. So there are two reactions really combined together, oxidation and decarboxylation. So what happens in your kuan class isocitrate. What happens first is the OH or the alcohol was, it was converted into a carbonyl. Do you follow? Therefore, oxidation. So, ka-remember mo, unsa nga coenzyme ang necessary when you convert alcohol to carbonyl? NAD or FAD? What is NAD? So therefore, this requires NAD. So in this process, NAD was converted to NADH because there is a conversion of alcohol to carbonyl. So after it's being converted into carbonyl class, what happens now? So musod na dahil ang atong gitawag na decarboxylation. In decarboxylation, one of the carboxyl group is removed. That's why it's called decarboxylation. You remove a carboxyl group. So when you remove a carboxyl group, it was converted into a carbon dioxide. So this is now the first step of the reaction nga na-achieve nato atong purpose, no? This is the first, di ba kay unsa gito atong purpose sa atuang kanang Krebs cycle? Ang acetyl ma-converted into carbon dioxide. So pag step 3, na-achieve na nato halfway ang ato ang purpose. Am I right? So we are already producing 1 carbon dioxide by decarboxylation of your isocitrate. Is that clear? Is that clear? Am I making sense, ladies and gentlemen? Hello? Nakagets ra ta ani class? Sige, so akong i-summarize ha. So gikan ta ni oxalo acetate ni combine siya ni acetyl. Am I right? So pila gani ni ka carbon si oxalo? It contains 4 carbons. Acetyl contains 2 carbons, producing now your what's the first product? You have your citrate containing how many carbons? That's containing 6 carbon. Okay, very good. So after that, citrate was converted to isocitrate. Once again, significance, why do we need to convert citrate into isocitrate? Because citrate cannot be oxidized, only isocitrate. Okay, so afterwards, because it's already suitable for oxidation, so isocitrate will be converted to alpha ketoglutarate. No? So this reaction is oxidative decarboxylation. So it's oxidation, therefore, it has the production of NADH. Correct? No, oxidation na siya. Oxidative decarboxylation. And because it's decarboxylation, there's also the production of carbon dioxide. So gitangtangan ni mo usa ka carbon. So if your isocitrate contains 6 carbons, how many carbons does your alpha ketoglutarate contain? It already contains 5 carbon because you remove 1 carbon dioxide. And that is already achieved when you reach the 3rd step of the Krebs cycle. Am I right? Am I right? So just like your step 1, step 3 is also a faucet. It's also a faucet in the Krebs cycle. Okay? So it's also a regulated step. So when you have plenty of NADH, no? So when you have plenty of NADH, it also can inhibit the step 3. It can stop, no? It can stop your step 3. However, aside from being inhibited class, it also has an activator. No, it also has an activator. So when you have plenty of ADP already, for example, no, nagamit ka og daghang kuan, nagamit ka daghang ATP. So ma-converted man na siya into ADP. So when you have plenty of ADP in your cell right now, so your cell will sense it, no? Specifically, kung daghan na kay kang ADP, mito say gamay naman ang imong ATP. So it will already, so it will actually make the step 3 faster, no? So it will activate step 3. Okay? So that's the beauty it. So pero siya gipapaspas ang flow ba if gamay na kag ATP. Is that clear? Is that clear? Am I making sense to you, everyone?
Yes, sir.
Okay. Sige. So that's it, no? So always remember that. Okay. So are we ready now? So let's proceed now to the fourth step. So the fourth step is another oxidative decarboxylation. So it's another oxidative decarboxylation. So it's the same principle at step 3. No? It's the same reaction as your step 3. Do you follow? So you still have oxidation, and then the oxidation is followed by a decarboxylation. But the thing about step 4, class, after the removal of the carboxyl group, no? After the removal of the carboxyl group, it will be attached with a coenzyme A. Do you follow? So, di ba? Kay kaganihan mang good, no? So, nakita ninyo ba sa atong step 3, nga after the removal of carbon dioxide, as is ra. Do you follow? But in step 4, after the removal of the carbon dioxide, ni-attach ngadto ang imuhang coenzyme A. So, there is an input of a coenzyme A. So, that's why nga the product actually contains succinyl coenzyme A. It's called succinyl coenzyme A. So as you can see, this is the succinyl part, class, no? The succinyl parts contain 4 carbons. Do you follow? So because can you remove naman sa carbon dioxide? Do you follow? But in addition to the succinyl group, it's attached already with a coenzyme A. So in this process, you're still producing NADH and you're also producing carbon dioxide. Is that clear? Is that clear? Class, am I making sense? Hello? Hello?
Sir.
Okay. Wala ni sila ni make sense ay. Balik. Oxaloacetate. Kalali, na wrong spelling ko. So you have oxaloacetate combines with your acetyl, forming now your coenzyme A, and that's just step 4. Forming now your citrate. Citrate is converted back to your what we call your isocitrate. Isocitrate undergoes oxidative decarboxylation, forming your alpha ketoglutarate. Unsay byproduct diri, class? Unsay byproduct sa conversion sa isocitrate? You have NADH and CO2. And then alpha ketoglutarate is converted into succinyl or succinyl CoA. So there's also a concomitant production of carbon dioxide and NADH. Do you follow? But the product is also attached with a coenzyme A. Do you follow? So that's as far as step 4 is concerned. So again, because you remove 1 carbon dioxide, you will expect to have that the next product is shorter with 1 carbon. So your alpha ketoglutarate originally contains 5 carbon. So the succinyl already contains 4 carbons. Diba? So it already contains 4 carbons. Is that clear? Is that clear? No? So that's step 4. Right? Sige. So let's now proceed to step 5. Dili lang ko na i-delete diha. So step 5 now. So, di ba? Na kay succinyl coenzyme A. Your succinyl coenzyme A contains a coenzyme A, correct? So in step 5, you're going to remove that coenzyme A. No, that's the step 5. That's the process of step 5. You're going to remove your coenzyme A. So when you remove the coenzyme A from succinyl, it's converted to succinate. And always remember, the removal or the breakdown of the coenzyme A is very, very energetic. Do you follow? No? The removal of coenzyme A releases a lot of energy. So the energy that is released by the removal of coenzyme A is used to drive the production of GDP to GTP. So guanosine diphosphate to guanosine triphosphate. So bali, kay daghan man kayo energy, so pag-release karon sa energy, gigamit siya to drive the production of GDP to GTP. So in the end, class, ang kanay mong GTP, it's chemically equivalent to 1 ATP. So therefore, in step 5, it's as if you're producing 1 ATP. Okay, the GTP that you produce here is chemically equivalent to 1 ATP. Do you follow? Do you follow? Do you follow? So, no, sa'yo naman kayo understand, no? So, step by step lang siya. So, pag abot ni step 5, gitangtang ang coenzyme A. You remove the coenzyme A. The removal of the coenzyme A releases energy. So, the energy that is released by this reaction is utilized to produce your ATP. Is that clear? Is that clear? Okay? So, therefore, from succinyl-CoA, it was converted now to succinate. Succinate. Okay? And then in the production of succinate, you produce 1 ATP. Do you follow? Do you follow? Okay? Sige. So let's now proceed to step 6. Okay, let's now proceed to step 6. Sige lang class, kay kanira yung curve cycle ang medyo taas-taas ko ano. The other pathways, we'll just consider them in brief. Okay? Sige. So let's now proceed
Sir,
to...
I have a question, sir.
Yes, Ben.
For stage 4, sir, it has an addition of CoA, but there's a reduction of carbon dioxide, sir.
Yes, that's correct. For step
I see.
4,
Thank you.
you remove the carbon dioxide, and then from the area where you remove the carbon dioxide, that is where the coenzyme A attaches. Okay, that's true.
I see. Thank you, sir.
So in step 5, gitangtang sa tunin mo ang iyong coenzyme A, no? So, right, kaya itong cell, no? Kaya ang removal ng sa coenzyme A releases energy. So para magamit to niya ang na-release ng energy. Okay, that's correct. Okay, so let's now proceed to step 6. So in step 6, di ba, nanatay succinate. So you're going now to oxidize your succinate. It's oxidized to fumarate. So the oxidation involves the conversion of single bond to double bond. Now, what coenzyme is needed when you convert single bond to double bond? You would need your, what coenzyme is that? FAD. You would need your FAD. That is why, as you could see there, no? If you try to look at there, what happened? No? So the FAD was converted to FADH2. So when you convert succinate to fumarate, what's the byproduct here? The byproduct is you get a FADH2. Do you follow? Do you follow? So that's it. No, that's the conversion there. Is that clear? Is that clear?
Yes, sir.
Okay. Sige. So nanti fumarate. Last 2 steps na lang yun. No? Sige. So at step 7, so you're going now to convert fumarate to malate. Okay? So there's no because there's no important process or byproduct here. Simply converting fumarate to malate. No? So I don't know what happens, pero gitangtangan na to og ano, gitangtangan na to og double bond, gibalik na sa nato o to single bond. Di ba? So ambot na nga itong cell. It's very, very fond of making fun of itself. No? So ni-remove siya og single bond kaya para ma-convert into double bond. But in the step 7, the double bond is regenerated back to single bond. Diba? And then, on the last step, malate is regenerated back to your oxaloacetate. So, in the conversion of malate to oxaloacetate, you got alcohol converted to carbonyl. So, you got an alcohol converted to carbonyl. So what is necessary if there's a conversion of alcohol to carbonyl? What is needed? You get NAD, no? So that is why in this process, the byproduct is NADH. Do you follow? You get NADH. So as you can see there, you will able to see that the original reactant, which was oxaloacetate, was regenerated back in the cycle. And then in the step 3 and the step 4, this is where you produce your carbon dioxide. So this is where we actually achieve the goal of the Krebs cycle. Am I right? Am I right? No? So in an initial class, when you exhale carbon dioxide, what do you think? No? What do you think when you inhale carbon dioxide? You think of the 3rd and the 4th step of the Krebs cycle, because that is where it is produced. So the carbon dioxide that you're exhaling right now is produced by the 3rd and the 4th step of the Krebs cycle. No? The conversion of isocitrate to alpha ketoglutarate and the conversion of alpha ketoglutarate to succinyl coenzyme A. Okay? That is where the carbon dioxide is produced. Okay? So that's it. That's already now your Krebs cycle. So as you could see, the acetyl of acetyl coenzyme A was converted into carbon dioxide by oxidation. And then at the same time, the original reactant, which was your oxaloacetate, was regenerated back. In the end, we produce, for every acetyl-CoA that enters, what do we produce? We produce 3 NADH, 1 FADH2, and 1 ATP. So these NADH and FADH will then undergo stage 4 of the biochemical energy production. Do you follow? Do you follow? So si NADH o si FADH2, sila na ang mo-proceed sa imuhang stage 4. Is that clear? Because they already contains electrons. They gained electrons already. And as we know, in the stage 4, it's called electron transport chain. So the electrons that they gained will be passed on there to produce your ATP. Is that clear? Is that clear? So diba, it's not very, very difficult to understand if your goal is to understand the steps, no? Kung sige mo kag-memorize without even knowing what does it mean, then you're like a monkey in a laboratory who keeps on memorizing. Am I right? Pero kung makasabot mo good kasi ang steps and then you understand the significance of these steps, you will appreciate it. And in fact, it would be easy for you to remember the different steps. Am I right? Am I right? I hope that I was able to share that with you, the excitement of knowing the Krebs cycle, because it's a very, very remarkable cycle. No, could you imagine that? Is that clear? Is that clear? Okay? Sige. So I actually presented a table kaya para di ko samot mo ka para ka nang dili lisod sa inyo ano. So I actually make a table. I think this is also in your notes that summarizes the different steps of the Krebs cycle. Okay? So you can actually scan through it if you want to memorize it that way, but I really suggest that you just memorize the cycle as it is. Do you follow? Do you follow? Okay, sige. So atong ikuan ha. Sige, atong sawayan kung ka-memorize sa bata without looking. Without looking there. Let's try. Let's challenge ourselves. Okay. Again, what's the first reactant of the cycle? The first reactant is your oxaloacetate. And then what happens? What happens in the first step? It will combine with acetyl, forming now your, what is formed? Citrate. Okay, very good. What enzyme is capable of doing this? You have your citrate synthase. Okay, very good. Afterwards, what happens now? Citrate cannot proceed to oxidation. Therefore, it would need to undergo rearrangement. So when it undergoes isomerization, it becomes what? It becomes isocitrate. Unsa gito nahitabo diri class? The tertiary
Tertiary to
alcohol must
secondary.
convert. Okay, very good. So what enzyme is capable of doing that? You have your
Aconitase.
aconitase. Very
Aconitase.
good. Aconitase. Okay. And then, di ba? So kung kasabot mo sa steps, madili ra listen i-memorize tas no? Sige. Then isocitrate will then undergo, what reaction will isocitrate undergo? Okay, ready naman siyang oxidize. So therefore, it will undergo oxidative phosphorylation. Oxidative decarboxylation. So when it does that, it will be forming your alpha ketoglutarate. Do you follow? It will be converted into alpha ketoglutarate. What is the byproduct in this step? You produce, of course,
NADH and CO2.
you produce first carbon dioxide, the carboxylation
Yes.
man. And it's oxidation involving the conversion of alcohol to carbonyl. Therefore, you get a
NADH.
What enzyme is capable of doing this? That is your isocitrate dehydrogenase. Okay? Again. Next, again, your alpha ketoglutarate undergoes another oxidative decarboxylation. So forming now your succinyl CoA. What happens here? What is the byproduct? You still produce carbon dioxide and NADH. Do you follow? And then the enzyme capable of doing this is your alpha ketoglutarate
Ketoglutarate.
dehydrogenase complex. Am I right? Am I right?
Yes, sir. Yes, sir.
So,
Yes, sir.
diba? So what's the next step? Kay naaman tay coenzyme A, unsa may atong buhaton ani atong coenzyme A? We remove it.
Removal of coenzyme.
So when you remove the coenzyme A, what happens?
Converted to succinate.
Very good. Oh, kamemorize sa si Ben, oh. It's converted to succinate when you remove the coenzyme A. So when you remove the coenzyme A, there is the production of a lot of energy. Am I right? So unsa may
Yes.
tabo? You produce GTP or
GDP
chemically
or GTP.
equivalent to ATP.
1 ATP.
Okay, very good. And then succinate will then be converted to fumarate. Unsa nga reaction ni naa kay carbon to carbon single bond, you convert it into carbon to carbon double bond. Therefore, this requires the production of, what is the byproduct? The byproduct is the production of
FAD.
FAD. Pero ang
FAD.
byproduct ay FADH2.
FADH2.
Okay? So by the way, what's the enzyme between succinyl CoA and succinate? This is your succinate synthetase. Succinyl CoA synthetase, right? Sakto ko? Please tell me.
Yes, sir.
Yes, sir.
How about here? This is your succinate dehydrogenase. Okay. Afterwards, fumarate is converted to L-malate. The enzyme responsible for this is your fumarase. And then L-malate is regenerated back to oxaloacetate. So in L-malate to oxaloacetate, you have alcohol converted to a carbonyl. So what is needed to convert alcohol to carbonyl? You would need your NADH. Okay?
NAD.
Very good. So that's the story of your Krebs cycle. Do you follow? Do you follow? Again, for every acetyl coenzyme A that enters, you get 1 FADH2, 1 ATP, and then 3 NADH. Kinsa tong makaproceed sa stage 4? Your stage 4
H.
and FADH2. Okay? So asa dire ang mga regulated steps? The regulated steps is step 1.
1 and 3.
Step 1 and step 3. Very good. So that's the story of your Krebs cycle. So it's not intimidating as it seemed to be, no? It's actually very enjoying to learn that. Am I right? Am I right? Question so far. Questions?
Sir, question.
Yeah. Christian.
So, then, sir.
Isabel, yeah. Go ahead.
So, the Krebs cycle is amphibolic, sir.
Yeah, but I will discuss that next meeting. Di na niyo ma-absorb. I will discuss that in the pre-recorded lectures. I will end my discussion the Krebs cycle alone. So I will discuss that later. To answer your question, yes, it's amphibolic. It's amphibolic. Okay, I will discuss later ng amphibolic. Okay? Sige. Yes, Christian.
May I clarify ra sir if kanang ni gain si electron transport chain og energy ni kuan sir no ni FADH og ni NAD.
Kaya diba naman si NADH o si FADH2 naman sila electrons. So ang kanang mga electrons sa imong FADH o NADH, murag bitaw magduwa kag bola. Nga for example, mag-catch ball mo. What do you notice kung mag-catch ball mo, di ba? Na-energy no nga imong ma-felt. So the same is true during electron transport chain because the electrons are being passed on from one protein to another. So the passing of electrons causes the gain of energy. So the energy that is gained by the electron transport chain will then fuel the production of your ATP. Okay? So that's the story. That's mo ginang ako iingon permitis ako estudyante. That's biochemistry in 10 seconds. Diba? That's biochemistry in 10 seconds. Okay? Sige. How about others? Any other questions? So again ha, i-memorize yung Krebs cycle ha. Memorize, kanila yun, no? So dili ko magpa-memorize ato mga glycolysis, Kivell, di mo ka memorize ato. Kay after all, ari naman sila paingon tanan. Di ba? Ari naman sila paingon. Okay? Sige. Any other questions? Any other questions? Yes?
None for me.
None. Okay, how about for others? None. Okay. So if none, then I will end my discussion here because it's been already 2 hours since I started talking. So for next meeting class, I will be, I mean, no longer next meeting. So for the pre-recorded lecture, I will discuss about the electron transport chain, and I also discuss the stage 1 and stage 2 of proteins, fats, and carbohydrates. Is that clear? Is that clear? Okay? So di ba mubura kay atong topic sa finals pa, you know? Kana ra kay para di na mo ma-stress. Wala na mo laing hunahunakon. Okay? So wala na yung questions? So yeah, we'll end our discussion here. Okay? Sige. So yeah, today we're gonna be